Chloroplast DNA. Mitochondrion, chloroplast, outer membrane, inner membrane, matrix, stroma, grana, DNA, ribosomes, thylakoid membranes. The endosymbiotic theory. Anaerobic eukaryotic cell DNA, alpha proteobacterium, aerobic endocytosis. Approximately 1 billion to 1.5 billion years ago, an anaerobic eukaryotic cell engulfed an aerobic eubacterial cell through endocytosis. The aerobic endosymbiote evolved into mitochondria. Likewise, endocytosis of a photosynthetic eubacterium led to the evolution of modern eukaryotic cells with mitochondria and chloroplasts. So, anaerobic eukaryotic cell DNA, alpha proteobacterium, aerobic endocytosis, evolution, endocytosis, cyanobacterium, mitochondrion, present day animal cell, then evolution, mitochondria, chloroplast, present day plant cell. Then chloroplast, organelle found in plant cells and eukaryotic algae, conduct photosynthesis. Chloroplasts are green chlorophyll pigment containing. Structure, chloroplast ultrastructure, outer membrane, intermediate space, inner membrane, stroma or aqueous fluid, thylakoid lumen inside of thylakoid membrane, granum, stack of thylakoids, thylakoid lamella, then starch, then ribosome, plastidial DNA, plastoglobule, drop of lipids. Plant cell chloroplast structure, outer membrane, inner membrane, stroma, lamellae, stroma, thylakoid, intermembrane space, and granum. Once again, stroma lamellae is their stroma region, granum, stack of thylakoids, intermembrane space there, thylakoids are attached. Then inner membrane, outer membrane, plant cell chloroplast structure. Some functions of chloroplast, lumen, thylakoid, stroma, etc. can be seen. Chloroplast, lumen region, thylakoid, stroma, light reaction. Chloroplast is the site for photosynthesis, light reaction. Thylakoid membranes, dark reaction. Stroma. Chloroplast is the site for photorespiration. Chloroplast is the site for photorespiration. So chloroplast genome, chloroplast DNA, cpDNA is also known as plastid DNA, circular double-stranded DNA molecule. Chloroplast genome, also known as plastid DNA, circular double-stranded DNA molecule. CT genomes are relatively larger, 140 kb in higher plants, 200 kb in lower eukaryotes. Multiple copies of genomes per organelle vary in size but are large enough to code 50 to 100 proteins as well as rRNAs and tRNAs. cpDNA regions include large single copy LSC, small single copy SSC regions, and inverted repeats, one IRA and IRB. Variation in length mainly due to presence of inverted repeat confers and a group of legumes back inverted repeats. LCS, IRA, IRB, SSC. Large single copy region, chloroplast DNA, Nicotiana tabacum. Small single copy region, inverted repeat B, inverted repeat A. Some chloroplast coding genes are there. Gene ATBB, RBCL, MATK, NDHF. Large single copy region of chloroplast, beta subunit of ATP synthesis, which functions in the synthesis of ATP via proton translocation. Large single copy region of chloroplast, large subunit of ribulose 1,4,5 biphosphate carboxylase oxygenase, Rubisco, which functions in the initial fixation of carbon dioxide in the dark reactions. Large single copy region of chloroplast, methylase, which functions in splicing type 2 introns from RNA transcripts. Small single copy region of chloroplast, subunit of chloroplast NADH dehydrogenase, which functions in converting NADH to NAD plus NH plus, driving various reactions of respiration. Then genome sequencing. Genome sequencing, 2 of the first CT genome sequenced. Liverwort, Mercantia polymorpha, tobacco, Nicotiana tabacum. Tobacco DNA is larger, contains 150 genes, and that of liverwort is 134. Basic genes, genes for rRNAs, tRNAs, ribosome proteins, polypeptide components of the photosynthesis system involved in capturing sunlight, catalytically active subunit of enzyme RUBP carboxylase, 4 subunits of chloroplast specific RNA polymerase, molecular systematics on cpDNA. cpDNA regions can be amplified by means of PCR. The resulted PCR products may be subjected to RFLP or DNA sequencing. Common cpDNA regions used in systematic study given. Chloroplast replication molecular mechanism is unknown. Independent of nuclear replication, plastosome copies are greatly amplified in mesophyll cells of the developing leaf and can reach values of more than 20,000 cells. The dual D-loop model for the initiation of chloroplast DNA replication, unidirectional elongation of nascent strand initiated from both origins, unidirectional fork movement toward each other, fusion of D-loops to crane type intermediates, bidirectional semi-discontinuous replication, then resulting daughter molecules, enzymes involved in ctDNA replication. These include DNA polymerase, DNA unwinding activity of helicase, primase, and or RNA polymerase, single stranded DNA binding protein, topoisomerase 1 and 2, origin recognition protein. Properties of ctDNA: non-Mendelian inheritance, self-replication, somatic segregation in plants inherited independently of nuclear genes, conservation rate of nucleotide substitution enables to resolve plant phylogenetic relationships at deep levels of evolution, familial level, mono dicotyledonous.